Okay, it's 1 minute after the hour. Thanks everybody for your precision on timing today, and we'll reconvene the meeting to begin the next session on hepatitis vaccines. Dr. Kevin Alt, chair of the hepatitis work group, will be providing an introduction and overview of today's session on behalf of the work group.
This is Kevin Alt, and I'll go through these slides relatively quickly to try to catch up a little bit. So as you can see from the title slide, we're going to be talking about a new vaccine, PreHevbrio. Next slide, please. 2022 is the 40th anniversary of the original hepatitis B vaccine, and we could talk about that quite a bit, but in the interest of time, I'll just say that the rate of new infections in the United States in the past 40 years has dropped 90% because of these vaccines. You all heard about our recommendations last year to expand adult immunization recommendations, and we're going to talk about this new FDA approval of the new vaccine. Next slide. And in the interest of time, I'm not going to mention all the subject matter experts, consultants, liaisons, ex officio members, and the voting members, but I would like to thank Dr. Wang quickly before we go on with the next talk.
Thank you, Dr. Alt. And we will move on to Dr. Francisco Diaz-Mitoma from Variation Biotechnologies, Inc., who will present on the safety and immunogenicity of this vaccine candidate. Thank you.
Thank you very much. On behalf of VBI Vaccines, thank you for the opportunity to be with you today to introduce the company and our 3-antigen hepatitis B vaccine, PreHevbrio. My name is Francisco Diaz-Mitoma. I am the Chief Medical Officer of VBI Vaccines. Next slide, please. As a new entrant to the US, we wanted to take a moment to introduce the company. VBI Vaccines is about technology company, and our first product is PreHevbrio, a recombinant hepatitis B vaccine approved by FDA, as Dr. Alt mentioned. And we have a pipeline composed of several prophylactic and therapeutic vaccine candidates targeting different infectious diseases and cancers. We're a global company with our corporate headquarters in Cambridge, Massachusetts, our research and development headquarters in Ottawa, Canada, and our fully owned manufacturing and research facility in Rehoboth, Israel, which is where the hepatitis B vaccine candidate is produced. Today, our presentation will be focusing on our 3-antigen hepatitis B vaccine candidate. Next. As we all know, hepatitis B remains a persistent US health problem that will require new solutions. There are numerous challenges with hepatitis B, including suboptimal surveillance, low awareness of infection, and that vaccine uptake rates have been persistently low, around 25%. Additionally, there has been an increase in acute cases of 11% in recent years, with changes in the incidence and epidemiology, with half of cases occurring in adults 30 to 49 years of age. Yet, despite these challenges, there is renewed focus on hepatitis B, with the committee's vote for the universal vaccine recommendation and broader surveillance guidelines. Additionally, the HHS has called for a 90% reduction in cases by 2030. To achieve these goals, new public health tools are needed, and it is my pleasure to introduce the main feature of our hepatitis B vaccine: the clinical development history, the safety and immunogenicity of the 2 phase 3 pivotal studies. And at the end of the presentation, I will also describe some key published legacy and investigator-initiated studies. Next slide. Before we start the description of the scientific differentiation of our vaccine, it is important to highlight the general features of the hepatitis B virus that are relevant to our vaccine. This pictorial is a description of the main components of the hepatitis B virus. As with other envelope viruses, it has 3 main components: its genetic material, a capsid or core, and the envelope. And the envelope has 3 distinct surface proteins: the small, the medium, and the large, which are shown in blue, green, and yellow. The pre-S proteins have a critical functional role in the recognition of the virus liver cell receptor. T-cell epitopes present in the pre-S domain elicit potent antibodies against the small f antigen, resulting in seroprotection against infection in hepatitis B susceptible populations. Next slide, please. This slide illustrates the features that differentiate our vaccine to those in the market. As I mentioned in the previous slide, the hepatitis B virus has 3 distinct surface proteins, small, medium, and large, known as S, PreS1, and PreS2. All available vaccines express the small S antigen. Our vaccine is the only one with a full set of envelope antigens, the small, medium, and large, which we believe leads to the potent immune response, resulting in increased seroprotection. The PreS proteins have a critical functional role in the recognition of hepatitis B virus liver cell receptors. We believe that the immune epitopes present in the PreS domains elicit a potent immune response that results in protection against infection. Also, PreHevbrio is the only one expressed in mammalian cells, as the other vaccines are expressed in yeast. It contains a common adjuvant, aluminum hydroxide, which has a good safety record and is also used in the standard of care vaccine. The adult dose contains 10 micrograms of antigen. So what is the significance of these differentiating features? Why does it matter? Next slide. The benefits are threefold. First, unlike currently available yeast-derived hepatitis B vaccines containing the small s, the antigens in our vaccine resemble the naturally occurring virion with similar glycosylation pattern and properly folded proteins representing linear and conformational B cell epitopes able to elicit efficient immunogenicity. Second, the full set of T and B cell epitopes of the viral envelope containing the PreS domains are more immunogenic than the small s. The PreS region is able to circumvent non-responsiveness to the small s antigen and induce a rapid and more potent antibody response. The efficiency of the PreS1 to elicit an immune response in non-responder mice highlights the additional benefit of a vaccine with these components. Several animal model studies support this finding. Both the 3-antigen conformation and the mammalian cell derivation confer benefits that result in an increased breadth of protection. And third, the PreS1 and PreS2 T helper and B cell epitopes result in high titers of antibodies that are required to prevent infection by all hepatitis B virus genotypes. Alterations in the A determinant of the small s surface antigen are largely responsible for escape mutants. The inclusion of the PreS region may reduce the risk of a better protect against vaccine escape mutants. This has been identified as a potential problem, especially in highly endemic jurisdictions. Next slide. This slide summarizes the real-world experience of our vaccine. In terms of US activity, the phase 3 program delivered 2 pivotal studies, PROTECT and CONSTANT, designed to achieve licensure in adults in the US, Europe, and Canada. Initiated at the end of 2017, and they were completed in 2020. In November 30th, as mentioned, the US FDA approved PreHevbrio for the prevention of infection caused by all known subtypes of hepatitis B virus in adults age 18 and older. And the American Medical Association current procedural terminology panel established a unique CPT code for the 3-antigen hepatitis B vaccine. Turning to the history of the vaccine, it was designed and initially produced at the Weizmann Institute. This is after the seminal work of Milich in the Scripps Clinic in the early 80s. And since 1989, 23 clinical studies have been performed in children and adults, resulting in the market authorization in 2000 in Israel, where it currently has preferential adult recommendations for certain patient subgroups. In Israel, it is licensed in 3 dose formulations, 2.5 micrograms, 5 micrograms for neonates, infants, and children, and the 10 microgram formulation, 1 milliliter for adolescents and adults. There has been a 20 microgram formulation that has also been evaluated in several clinical studies. Approximately 750,000 people have received the vaccine in Israel. Next slide. The phase 3 clinical program comprised 2 pivotal studies, PROTECT and CONSTANT, which look at immunogenicity and safety in comparison to the most frequently used standard of care vaccine, Engerix-B. The PROTECT study was published in Lancet Infectious Diseases in May of 2021, and the CONSTANT was published by the JAMA Network in October 11, 2021. Next slide. The first study, PROTECT, is a multi-center, double-blind, randomized control study conducted in 28 community clinics and academic hospitals in the US, Finland, Canada, and Belgium. And the study enrolled 1,607 participants with an age range of 18 to 90 years of age. The co-primary endpoints are non-inferiority in adults age 18 and older, and superiority in adults 45 and older. Secondary endpoints included safety and antibody concentration, as well as kinetics of the seroprotection rate. The CONSTANT study is a double-blind lot consistency study with a controlled vaccine arm. The study end size was expanded to complete the safety database requirement. Randomization was to 4 arms, with 1 arm receiving the control vaccine and 3 arms receiving the candidate vaccine, with a total of 2,800 participants. The age range was 18 to 45. And the primary endpoint was the geometric mean concentration of antibody across the 3 vaccine lots. Secondary endpoints were safety and seroprotection rates. Next slide, please. This is the breakdown and subgroup distribution for the studies. In PROTECT, 82% of the age cohort was older than 45. In PROTECT, we enrolled 43% of participants in the US, and in this US-based population, we strove to ensure racial and ethnic diversity. And we saw good representation of African Americans as well as Latinx. We also enrolled 40% in Europe and 16% in Canada. 95% of participants completed the study. In the CONSTANT study, we enrolled 2,838 participants, with 27% in the US and 70% in Europe. There were more participants from Europe, especially Finland, as in Finland, hepatitis B universal pediatric vaccination was only recommended in 2016, allowing us to enroll a younger age cohort of 18 to 45. All volunteers in this study were healthy, and all potential participants with chronic health conditions were excluded, including people with body mass index higher than 30. Next slide. A critical endpoint of the pivotal program was to confirm the safety of the 3 antigen vaccine observed in previous clinical studies. Next slide. The integrated safety analysis demonstrated good tolerability and no unexpected reactogenicity. Our vaccine was associated with increased rates of mild to moderate injection site pain, tenderness, and myalgia. This reactogenicity generally resolved without intervention in 1 to 2 days. There was no increase in reactogenicity over the 3-dose vaccine schedule, and there were low rates of vaccine discontinuation. Next slide. The rate of unsolicited adverse events was similar in both vaccine arms. There were no unexpected safety signals, and there were no unusual patterns or concerning clusters of severe adverse events. There was no increased rate of medically attended adverse events or new onset of chronic illness in comparison to the controlled vaccine arm. Next slide. Summarizing the safety data, there was a high completion rate in both studies, and the most common finding was local reactogenicity that resolved with no intervention. There was no difference in medically attended adverse events between vaccine groups. Severe adverse events were uncommon with both vaccines, and there was no clustering of adverse events. There were 2 severe adverse events reported as possibly related, a severe gastroenteritis and an ankylosing or tongue tie, that upon independent review, it was determined that there was no associated causality. There was one death in the constant study to a participant randomized to the PreHevbrio arm. The autopsy ruled the death was due to a pre-existing hypertrophic heart disease. Next slide, please. The PROTECT and CONSTANT studies analyzed the immunogenicity of our vaccine and the standard of care control. Next slide. The co-primary endpoints were met 4 weeks after the third vaccination. These co-primary endpoints were based on the comparison between arms of the seroprotection rate represented by 10 milli-international units per milliliter of antibodies against the surface antigen. That's represented in the x-axis in this slide. And this represents a surrogate of protection against infection. In the left panel on this slide, the first endpoint, non-inferiority in adults of all ages, was demonstrated with a between group difference of 14.9% in the per protocol set in all adults. This difference was sustained and slightly increased in the second endpoint, and statistical and clinical superiority of PreHevbrio over Engerix was demonstrated in older adults with a between group difference of 16% in the full analysis set. Next slide. Now, I would like to draw your attention to the center panel. The forest plot demonstrates the difference in seroprotection rate between subgroups of interest, which are listed by age cohort, by diabetes diagnosis, or by body mass index in the left column. The vertical broken orange line is the null hypothesis, or zero seroprotection difference. To the left, it favors the control vaccine, and to the right, it favors PreHevbrio. The diamonds are the mean difference in seroprotection, and the horizontal lines are the 95% confidence interval. The right panel is the antibody geometric mean concentration, with our vaccine demonstrating 4 to 8-fold increase in the antibody concentration over the control. This improvement was sustained regardless of age, diabetes, or body mass index. Next slide. There are additional analyses showing that the improvement in seroprotection and antibody concentration was also sustained regardless of alcohol consumption, smoking, as well as gender. Next slide. And in this table, you can see it was sustained regardless of race, ethnicity, or geographic jurisdiction. Next slide. The blue columns in this slide show the higher seroprotection rates that were achieved with PreHevbrio after 2 and 3 doses in comparison to the control. In PROTECT, the difference after 2 doses was pronounced with 87% versus 39% seroprotection rate. And in CONSTANT, PreHevbrio also achieved higher levels of seroprotection after 2 doses than the control, 90 versus 51%. Next slide. Moreover, the seroprotection rate was higher for PreHevbrio than the control at all time points and in all age cohorts. The seroprotection rate was higher after 1 dose and more than double after 2 doses with our vaccine than with the control vaccine. In addition, the 12-month durability of seroprotection was also higher with PreHevbrio than with the control in all adults. Next slide. We just reviewed the seroprotection kinetics. Now we are seeing the kinetics of antibody titers. These are the immunogenicity results of the CONSTANT study, demonstrating a rapid and consistently high rate of seroprotection and higher concentration of antibodies that were achieved with PreHevbrio in young adults when compared to the control vaccine. The primary endpoint of load consistency was met. Our vaccine, next slide. Our vaccine has extensive real-world experience. We want to highlight 3 investigator-initiated studies in populations where there is particular concern on low or non-response to Hep B vaccines, and how PreHevbrio is able to circumvent non-response in the majority of patients. These studies are published in refereed journals. Excuse me. Next slide. In these three published studies, the benefits of a more potent hepatitis B vaccine was seen. Patients with end-stage renal disease, or HIV, are particularly at risk of hepatitis B virus exposure, and the standard of care requires frequent serology testing. And often there is low seroprotection after standard of care vaccination. In the renal failure study, these patients did not respond to a double dose of Engerix-B, but after 3 doses of our vaccine, a seroprotection rate of 86% was achieved, higher than the standard of care and historical control of 56%. Similarly, in the HIV positive patients, a high seroprotection rate was observed after 3-dose schedule of the 3-antigen vaccine of 84% versus historical controls of 17 to 53%. And in the third study, adults who did not respond to more than 3 doses of a conventional yeast-derived vaccine, the seroprotection rate was 87% after 1 dose of our vaccine. Next slide. To summarize the information presented today, next slide. The safety profile is comparable to Engerix-B and consistent with the 20-year known history and usage of our 3-antigen hepatitis B vaccine. There are higher rates of protection in all adults and robust immunogenicity across age cohorts. In particular, for older adults where immunosenescence is a concern, we saw an improved response in our studies for people 45 years of age and older, and for people 65 years of age and older. There was a rapid onset of protection for all ages and all time points. In particular, I wanted to note the 90% of participants in the study, 18 to 45, reached seroprotection after 2 doses. This population is of increased concern as nearly half of all cases of Hep B are occurring in this age group. And finally, there is improved immunogenicity in key high-risk populations. Those with chronic conditions often have a suboptimal response to vaccines, but there we saw improved seroprotection for people with both diabetes and obesity. There is a renewed focus on hepatitis B with both new immunization and screening guidelines. These efforts will require new approaches, and we believe our vaccine is a public health tool to achieve these goals. Thank you very much for your time and consideration, and we look forward to continuing this process. And I would be happy to take any questions at this time.
Thank you very much. This presentation is now open for questions. Dr. Alt.
Since you have 20 years of experience with this vaccine, I wanted to know if you had any data specific to pregnancy.
We had a handful of pregnancies in the study, and we didn't see any adverse events during the study. As part of our post-licensure commitment, we're developing a protocol now that we're presenting and submitting to FDA as a pregnancy registry. But otherwise, it's a very limited experience that we have with pregnancy.
Thank you.
Mr. Sanchez?
Thank you. Very impressive results, I have to admit. And it seems like, at least from your presentation, that it's also been used in neonates and currently being used in Israel in neonates and infants. But my question really, a little bit more about these surface antigen, the pre-S1, pre-S2, and of course the hepatitis B surface antigen. Are these 3 distinct different proteins, or when I guess I'm used to the pre-fusion of the RSV and that it changes the format as it attaches, or are but from your schematic structure, it seems like there's 3 distinct ones, or is this one that changes their shape in relation to when it infects the host?
Thank you very much, Dr. Sanchez, for the question. That's really one of the main differentiating features of our vaccine. So what happens in the PreS1, PreS2, and S, when you look at the total length of the PreS1, which is the largest protein, is a protein that has something like 226 amino acids in length. And then there is an overlapping region. There is a unique region that is about 100 amino acids. And there is a common region for the small s. Similarly, with PreS2, there is a very specific domain that is around 50 amino acids that is very specific for the PreS2 domain. So, and the characteristics of these PreS domains is that they contain very potent T cell helper cytotoxic as well as B cell epitopes. Therefore, they enhance immunogenicity.
So there are three distinct proteins altogether on the surface.
Yes, correct.
Not just conformational differences.
Not just conformational differences. They are very distinct with the PreS1 domain and the PreS2 domain are very distinct, and they are not contained in the small s.
Thank you.
You're welcome.
And then what is your experience with the neonatal in Israel?
Well, the vaccine has been used as a birth vaccine for the last 20 years, and it continues to be used in neonates and in children. So it has been extensively used in pediatrics. And, you know, when you look back at the legacy studies, we had 23 clinical studies besides the pivotal phase 3 studies that we conducted. There is 23, and actually 12 of them are in neonates or children. So there is extensive literature about the use of this vaccine in Israel.
And do you plan on getting an FDA approval for newborn vaccination?
Well, we currently have a waiver from FDA for the pediatric indication, but we remain open to further develop these. Especially there is interest from investigators to use it in populations where there is a need for an enhanced hepatitis B vaccine.
Thank you. We'll move on to Dr. Sanayas.
Thank you for that presentation. You mentioned that in Israel, the vaccine.
Oh, Dr.
Sorry.
We lost you at the very end. Could you please repeat your question?
I'm sorry. You mentioned that the vaccine was recommended for certain groups in Israel. Could you tell us what those groups are?
Yes. So it has preferential use for healthcare workers, first-time responders, as well as end-stage renal disease and HIV positive and immunosuppression for patients who have immunosuppression.
Thank you. Dr. Cotton.
Yes, regarding patients on immunosuppression, so, you know, the data look pretty good for ESRD and HIV, but are there actually subgroups with, you know, organ transplant, of which there are a lot in Israel and elsewhere, certainly, and other much more immunocompromised patients?
There has been very small investigator-initiated studies in transplant recipients, especially very profound immunosuppression, like liver transplants, where the results have also been positive. And in those particular studies, the higher doses have been used, as I mentioned, the 20 micrograms formulation has been used.
Thank you.
What about,
and any chance you have any data in patients getting things like rituximab or any history of rituximab or some of those really challenging patients?
That's very limited information on those particular groups.
Thank you.
You're welcome.
Thank you. Are there any additional questions? one more question for you. And I don't know how much available post-market safety surveillance data there are from other countries. Is that something that you have access to or that countries share with you?
The main observations that we have is from the periodic safety reports that are submitted every 2-3 years to the Ministry of Health in Israel. And in those reports over the last 15-20 years, we have not seen any clustering of severe adverse events or anything like that.
Thank you. And can I clarify, does the, and either Dr. Alt or someone else can answer, but just to clarify, for the FDA approval, does it include pregnant women, exclude them, or not make any statement about pregnant women?
Is not indicated for pregnancy. It excludes pregnancy.
Okay, thank you.
Yes.
I was thinking about when we update the adult immunization, we'll have to put a footnote in there, but that's not till next year, so we'll be fine.
Okay, that seems like decades away at this point. Okay, great. Any other questions? Wonderful. I think we'll go ahead and close this session. We are actually catching up a little bit on time. If we can take another 5-minute break, and then we'll reconvene. So 11:38 on West Coast time, 2:38 East Coast time. Thanks, everyone.